So good morning, students. So today we'll see translation. So let me take the slide first. Okay. So translation is, so before translation, I have to say, 1 minute. So before translation, I have to say what is all other thing like central dogma. Okay. So let me explain that first. Okay, so the central dogma. So let me explain here. Central dogma. Central dogma means, so first replication, okay, transcription and translation. Okay, translation. So replication is nothing but, so DNA will be formed from previously formed DNA. Okay, so the DNA's replication is the exact copies of the previous DNA. Okay, so transcription is nothing but, so mRNA is produced from DNA. So production of mRNA from DNA. Okay, so that is transcription. So what is translation is, so from mRNA proteins are formed. Proteins are formed from mRNA. Okay, so those are called translation. So we'll directly dive into translation. Yes. So for translation, so this is an important category you have to know. Okay, so that is genetic code. So the genetic code is nothing but they have this, it's a triplet codon. Okay, actually, so you can see, so genetic codon means, so it's a set of certain rules. So which is in triplet codon, that is in triples. Okay, so okay, you can see this table. So it will be in three, that is UUU, UUC, UUA. Okay, so these are some triplets. So each triplets will be coding an amino acid. Okay, and so amino acids will code more than one triplet codons. Okay, so in that, so there are many amino acids are there which coded by codes, triplet codes. So the important thing is AUG. So AUG is the start codon. It's called as initiation codon or start codon. So it codes methionine. So it's written as met. So met is nothing but it stands for methionine. Okay. Yes. AUG. So the process translation will happen, will start only if this particular AUG, the codon AUG comes. Okay, only if the AUG is there, so it will start its in translation process. So what about this UAA, UAG and UGA? So these are stop codons. So stop codons, whenever the continuous process will happen, so which we'll see in 2 minutes. So after this, some few process of amino acid formation. So if case of any amino acids, like maybe triplet codons like UAA, UAG, UGA, any one of these comes to the site of translation, what happens? Codon, that is the process, will stop. Okay, so because they are called as stop codons. So these three codons is there only to stop the process. That is stop codons. Okay. Okay, fine. Okay, we'll see. So these are some properties of genetic codon. Okay, triplet code. That is, I told you that it's only in three forms. Okay, non-ambiguous and universal. Okay, non-ambiguous means they can code. Okay, so they can, so I mean specific codon will be having only one amino acid, but one amino acid can be coded by, yes, yes. So one amino acid can be coded by many codons. See, so in this picture you can see this fetal aldehyde can code. So these amino acid, they can code UUU, UUC. So they can coded by more than one triplets, but one triplets will be having one amino acid. So UAE will be coded only by one amino acid, leucine. LEU is leucine. UAE is coded by leucine. Okay, but this leucine can be coded by more than one triplet, that is UA, UUA, UUG, CUU, okay, etc. So these are coded by, okay, one amino acids can be coded by many triplets, but one triplet will be having that is one genetic code. Okay, one code will be having only one amino acid. Okay, so these are the properties of non-ambiguous and degeneration. Okay, it's non-overlapping. So if there is any mutation occurs, whether if the sequence is inserted or deleted, anything. Okay, so there won't be any overlapping. So it will take only in three forms, that is in triplet forms. Okay, so there is no punctuation, there is no stop in between. Okay, genetic code. Okay, so that is one thing I told. Start and stop codons. I told already. AUG is start codon, which also codes for methionine, and it will be coding for formyl methionine in prokaryotes. Okay, what about this UAG, UGA, and UAGs? Okay, so these are stop codon or termination codons. Okay, they will be there only for stopping the process. Polarity is, so they can code only the direction 5 dash to 3 dash. So we'll see the process directly. Okay. Okay, so there are some factors which involves in initiation. Okay, so the initiation factors IF1, IF2, and IF3. Elongation factor EF, TS, TU, then EFG. Then releasing factor or termination factor. Okay, RF1, RF2, and RF3. Okay, fine. Okay, then this thing. So we'll see steps directly. So we'll see the outline. Okay, steps of translation. What happens? So first, the mRNA is getting attached. Okay, so the main thing is, so these are amino acids. Okay, so these color balls are amino acids. Okay, so the mRNA is first attached to the part of ribosome. So which is the mRNA here? So this part is mRNA, which is formed from DNA, because this is mRNA. Okay, so mRNA was attached to the ribosome. So I will show one more picture. Yes. So this is mRNA, and this big bubble-like structure is called rRNA or ribosome. Okay, this is ribosome. The first step is attachment of mRNA with ribosomes. Okay, next tRNA that carries amino acids. Okay, so this is tRNA. So it carries these amino acids, will match the triplet codons of the mRNA. See, you can see. So let me zoom and show you. Okay, so UUC is there in mRNA. So what will be the complementary pair? AAG. Okay, so this particular tRNA with this anticodon AAG, okay, it has the amino acid. Okay, so they carry the amino acid along with them. So they come and attach, and all these amino acids are getting attached to form a protein or polypeptide chain. Okay, so this is the second point. The third point will be the amino acid chains become longer. So that's what I told. So longer means it will produce all the polypeptide chain or protein. Okay. Yes, so you can see in this pictures. So three pictures I have. See, this is mRNA which is formed from transcription process. Okay, these are amino acids taken by this tRNA. So there are two sites called P site and A site. Okay, so this P site, okay, so they have, so that is the first site. Okay, so then it's covered, I mean it's followed by A site. Okay, so there are two room like structure. Okay, these amino acids get attached. Okay, so gets attached to form a peptide chain. Finally it forms a full protein. Okay, so these are the steps. The first one is activation of amino acids. Okay, so activation of, so amino acid first it has to be activated. Only then it can form a full protein, a matured protein. Okay. Okay, with activation of amino acid is by amino acid tRNA synthase. Okay, so amino acid, so it's by amino acid enzyme complex. Okay, so AMP is adenosine monophosphate. Okay, so with the help of energy, the amino acid is getting activated. Okay, so this is the thing. Second thing is initiation process. Initiation process where the mRNA gets bind to the ribosome, where the particular ribosome contain a large subunit and small subunit, you know, right? So they mRNA get attached to the small subunit. Okay. Okay. So the third one, I mean, second process of this translation is elongation. So then elongation process, what is happening? I told you, right? There are sites or there that is a room-like structures. Okay. So first it is initiated by AUG, it is start codon. Okay. It also codes for amino acid methionine. Okay. You know, right? So the process is continuously happening with the help of elongation process. That is the TRT will carry the anticodons along with amino acids, okay, which is complementary to mRNA, the codons present in mRNA. Okay, they carry the amino acids. See, you can see there is the elongation of amino acid chain. See, okay, can see, right? So these steps will continuously happens. Okay, the last one will be termination process. This termination process, as I told you, this process will happen until these stop codons arise there. So stop codons UAA, UAG, and UGA. So any of these stop codons, any of these three, or if anything comes out of these three, so the process will stop. The termination process will happen. Okay. So once the termination has happened, so the peptide, that is protein chain, will get detached from the ribosome complex. Okay, a Faraday ribosome complex, it will detach from those complex. Okay, so that will be happening with the help of these releasing factors. Okay, so okay, fine. So post-translation modification will be, so the protein modification, that is protein alignment or protein structural modification of protein will happen after the formation of protein. Okay, okay, fine. So these are the different steps happening in translation process. Okay, so hope you guys understand. We'll see the next class. Thank you.